Today we are going to be talking about Chapter 6, Section 3, and this is mostly about Mendel's experiments and what he learned from them. So we know that Gregor Mendel studied genetics, and his organism of choice was the pea plant. So what he did was he used pollen from specific plants to fertilize other plants, and the way that he chose those two parent plants is that he always went for the purebred version of the plant. So I'll be using flower color as an example here. He had in his garden purebred purple flowered plants, which meant for generation after generation, he only bred together those purple flowered plants so that he was sure that their offspring were always going to produce purple flowers. He also had a purebred version of a white flowered plant. Same thing. He would only breed together white flowered plants for generation after generation, producing a purebred white flowered parent. Those two plants, the purebred purple and the purebred white flowered plant, those were the pea generation. We use P for parent. So the two parents were the purebred purple and the purebred white flowered plants. He crossed them together and produced a generation that we call F1. So that's the children of the original two parent plants. Now, the way he did this, he had to be very careful. He was doing scientific experiments. He wanted to make sure that he only pollinated a plant with the pollen from the other purebred plant. Peas can actually self-pollinate themselves. If you look in a pea plant flower, there's a female part, and the pollen would stick to this top of the female part in order to fertilize the ovules. Those are essentially plant egg cells in there. And then they would grow into the peas in a pea pod that you would see later on. But all of these other little parts, those are the male pollen-producing parts. And he didn't want pollen from this purple plant to get on its female part, and essentially that would be this plant fertilizing itself. He didn't want that to happen. He wanted this plant to be fertilized with the pollen from a white flowered plant. So by cutting off all of these pollen-producing male parts, the entire flower can still live. The female part is perfectly intact. So he would use a paintbrush to get pollen off of a white flowered plant and place it directly on the female part of this purple flowered plant. So the two plants, the purple and white flowered plants, were the pea generation. He selectively bred them to produce their children, which is the F1 generation. Now, he allowed those F1 plants to self-pollinate. So that means all of the plants produced from that original cross, he allowed them to pollinate themselves. And then their children, or the grandchildren of the original two plants, is called the F2 generation. So the interesting thing that Mendel noticed is in the F1 generation, that was the first set of offspring from those original parents, all of the plants had purple flowers. Even though one parent was purple and one parent was white, all of their offspring were purple. And so that was something interesting that Mendel noted. So then he really wanted to do that next part of the experiment and see, okay, well, all of these F1 plants are purple. What if I let them self-pollinate? What will the F2 generation look like? And we know today that all of those F1 plants were heterozygous. He didn't know that at the time because, remember, he didn't know really anything about genetics. None of this had been established before he did his experiments. But today we know those F1 plants were heterozygous, so we can already kind of think to ourselves that we might see a different color combination in that F2 generation compared to what we saw in F1. And that's exactly what Mendel noticed. He saw that in the F2 generation, some plants were purple and some were white. So here's his experiment summarized in this really handy diagram. Two purebred parents, purple and white, were crossed together to make these offspring. We know that these are heterozygous. Mendel called them hybrids because they were a mix of two purebred plants. So today, whenever you see the word hybrid, that means the same as heterozygous. So he allowed these hybrids to self-pollinate. So this one flower, its male gamete in the pollen landed on the female part of the flower and produced seeds. And in the F2 generation that resulted, there was a 3-to-1 ratio, or about 75% of the offspring had purple color, just like the F1 generation did, but 25% of the offspring had that white color that was not seen at all in the F1 generation, but it was present in this second parent. So that was really interesting for Mendel and allowed him to come up with some very foundational ideas about genetics that we still use to this day. The patterns that he noticed were true across all of these different characteristics. So we talked about flower color, but look at these other characteristics about the shape and color of the peas or the shape and the color of the pod. No matter what characteristic he was studying, he always saw a 3-to-1 or approximate. Remember, this is actual real science, so it's not going to come out to be a perfect number, but it was always approximately a 3-to-1 ratio of dominant phenotype to recessive phenotype. And this is how Mendel actually figured out what the dominant and the recessive phenotypes were. If we go back and look at that picture, two purebred parents, when you cross them, whatever your phenotype is of your F1 generation will have to be the dominant phenotype. And this one that got covered up was the recessive phenotype. So these discoveries showed that blending hypothesis was definitely false. If it were true, we would just get lighter and lighter purple plants in each generation, but that's definitely not what Mendel saw. So he took his results and came up with 3 laws, and we call these Mendel's laws, and they're the foundation of our understanding of classical genetics, sometimes just called Mendelian genetics, because he came up with the foundation of our understanding. The first is the law of dominance. The second is the law of segregation. And the third is the law of independent assortment. So we will spend the rest of this presentation talking about each one of those laws and what exactly that means and how it relates to what we previously learned about meiosis. The first one is the most straightforward. This is the law of dominance. We actually have already discussed this just in looking at the results of that F1 generation. The law of dominance tells us when you cross 2 purebred individuals that have different versions of a trait, all of their offspring will exhibit whichever trait is dominant. And this is where the term dominant and recessive comes from. Mendel didn't know about genes and alleles, but he could view a phenotype, and whatever phenotype showed up in the F1 generation, he just called that the dominant one because it was covering up the second phenotype, and he called that second phenotype, the one that disappeared in the F1 generation, recessive. Now today, in our modern understanding, we do know about chromosomes and genes and alleles. So one way that we would think about this today is that organisms that have a heterozygous genotype, remember that would be having one dominant allele and one recessive allele. If they're heterozygous, they will always have the phenotype of the allele that is dominant. Another way to think about that is that the dominant allele, you only have to inherit one of them in order to show the dominant phenotype. But if you were going to show a recessive phenotype, you would have to inherit 2 recessive alleles to show that phenotype. So again, our example here was with flower color. We had the purebred purple and white flowers. In the F1 generation, all of the offspring have the purple phenotype, therefore purple must be the dominant allele. Our second law is the law of segregation. And in the law of segregation, this has to do with when the gametes are made. Those reproductive cells, which are the gametes, remember they only have one copy of each chromosome. So in a gamete, it will only have one or the other of the two alleles from the parent that made that gamete. This is because those alleles separate when those homologous chromosomes become separated from each other during anaphase 1 of meiosis. So let's look at an example of what that actually means. In pea plants, there is a trait of having tall stems versus short. So we're going to use the letter T for this gene for plant height. Capital T would be a tall plant, lowercase t would be a short plant. And in this Punnett square, we're looking at one parent, capital T, lowercase t, that means they are heterozygous. The second parent, also capital T, lowercase t, that is a heterozygous genotype. So if we look at both parents, when they produce gametes, this capital T could be in a gamete or this lowercase t, but this parent is never going to produce a gamete that has both copies of the alleles in it. So the law of segregation is basically demonstrated every time you draw a Punnett square. This parent is either going to pass down one allele or the other. These two alleles will separate. Segregation means separation. So those two alleles will separate from each other, one going into one gamete, one going into the other. And our Punnett square is telling us that the offspring could either inherit the gamete with this allele or the gamete with this allele, but it can't inherit both of these alleles that came from the same parent. And that is what our law of segregation tells us. Finally, we have the law of independent assortment. And in this law, we're talking about alleles for different characteristics being inherited separately from each other. And that means that a pea plant, whatever allele that it inherits for flower color, is not going to impact the allele that it inherits for flower position or the shape of its seed or its height and so on and so forth. So essentially, you can have either purple or white flowers and have either yellow or green seeds, or you could have tall stems or short stems. Having a dominant allele at one gene position does not affect what kind of allele you will inherit at a separate gene position. This is because of the way the homologous pairs line up in metaphase 1. We will take a look at a picture of that on the next slide just to relate that to the law of independent assortment. But before we do that, let's revisit genes and chromosomes. These are the 7 chromosomes that are found in pea plants, and the gene locations are marked on there. So we see that a gene for flower color is on a completely different chromosome than the gene for the shape of its pod or the color of its pod. So let's relate that to what we know about meiosis. In meiosis, remember that those homologous chromosomes line up in the center of the cell during metaphase 1. We have the blue chromosomes that represent what got inherited from the father, and the red ones represent what were inherited from the mother. So it is possible that as they line up, all of the father's chromosomes lined up on the left, and the ones that were inherited from the mother lined up on the right. But it is equally possible that we would see this possibility too over here, where you see the blue chromosome on the left for this top pair, but now the blue chromosome is on the right in the bottom pair. Now these chromosomes, they are lining up independently of each other, and that's why this is called independent assortment. So it's just as likely for them to line up this way as it is this way. But when you look at the end result for both of these two possibilities, you can see we get 4 completely different combinations in the gametes that are produced. And that is where independent assortment comes from. Just because you inherit the large blue chromosome doesn't mean you also have to inherit the small blue one. You could get the large blue one and the small red one. So hopefully this is helpful in understanding the law of independent assortment. If you still have questions, please make sure that you ask those in class.